My name is Cristina. I'm a master's student with work in VAS Center of Cognition, Brain and Language in San Sebastian. And I'm going to talk about, as I said, mapping cerebrovascular activity and vascular lag mapping in glioma with multi-echo fMRI. The first, cerebrovascular activity is a physiological response in which cerebral blood flow is regulated in response to a vasoactive stimulus. This induces changes in arterial CO2 pressure, causing vasodilatation. The vasoactive stimulus can be produced by administration of intravenous acetaminophen, hypercapnic challenge, or no external gas manipulation, such as resting state or breath hold. CVR mapping is a very useful technique not only to monitor health, brain, but also to vascular health disorders, such as stroke and aphasia, hypertension, neurodegenerative diseases, sleep apnea, and brain tumors, such as gliomas. Are diffusely infiltrating tumors that originate by glial cells. Alterate the relationship between neurometabolic activity and vascular response, which is called neurovascular component. Neurovascular component can be a critical factor in the use of fMRI in procedural mapping, as can lead to false negative activation due to the absence of normal vascular response, such as cerebral blood flow. This vascular response can be measured by the means of CVR mapping. However, can CVR mapping be used in the procedural mapping of abnormal vascular response in glioma with fMRI? Some studies have shown that CVR mapping in glioma can be measured by hypercapnic challenge. All of these studies has used a single echo fMRI and an hypercapnic challenge using the respirator. One of them is Panfiesta and the colleagues were measured a CVR impairment and distribution throughout the brain. And we could see the results. We see that in the right side, in the C, there's a decrease in the CVR comparing to in the glioma. And in the first one is a healthy subject. There's other studies that also measure the CVR mapping in glioma. Mucas and their colleagues investigate more about the peritoneal areas, where he could see that there's an hemodynamic change that correlates with the tissues, the glioma size. And also, CBOOK did a study about the infiltration beyond the visible areas, and that we can see in the tumor areas. And for last, Mucas in 2021 also did a study about the CVR patterns in brain radiation process. However, not all of the people can tolerate this gas manipulation and doesn't have this equipment of the respirator. So there's other ones that did a breath hold test to have this hypercapnic challenge. Also in common that had single echo fMRI. In 2011, Pillay and Saka evaluated the neurovascular component associated with low-grade gliomas using CVR and breath hold. And Irambu in 2016 also measured the CVR maps, where we could see the results that he got a decreased CVR in the tumor area. So this can see that with a breath hold test, we can have a decreased CVR in the glioma. However, we know that a fMRI is sensitive to noise and artifact. And it's in the study by Moya in 2021 tell us the benefits of the multi-echo fMRI with breath hold, using a multi-echo ICA diagnostic technique where he compared the aggressive technique to a conservative more approach. In this case, he used a general linear model where they evaluate as a dependent component the fMRI signal, and as an independent component the PET CO2 convolve the hemodynamic response function. That they can be at the transit delay between the measurements of the measurements delay. There can be also a delay in the signal between the vascular transit and also the vasodilatory. To take this into account, we use a lag general linear model. They shifted the PET CO2 HRF 60 times to take this measure. the results, as I explained before, it did a denoising technique, where they compare a aggressive, moderate, and conservative approach. In this first image, we can see the results of the CVR maps and the vascular delay maps in single echo. Then the multi-echo aggressive and the multi-echo conservative. And we can result that the multi-echo conservative approach is the optimal for CVR maps. This multi-echo conservative approach took into account the ICA rejected component that were orthogonalized with respect with the lag PET CO2 and the ICA accepted components. Here we can see the ICC maps of the CVR and vascular delay, comparing it with the single echo, multi-echo aggressive, and conservative. In the histograms, we could see that the most reliability is in a multi-echo conservative approach. The goals of the study is part of the prosthetic mapping project in BCL and the Hospital of Cruces in Bilbao. The main objective was to see that the breath-hold-induced CVR mapping with multi-echo fMRI to improve the accuracy and the durability of the CVR maps and vascular lags in glioma patients to evaluate the brain's vascular health. The hypotheses were that one of them, that the implementation of the conservative approach by Moya in glioma will deliver a more precise mapping and vascular lag maps, minimizing the influence of motion artifacts. Also, that there will be a decrease of CVR values in tumor-affected areas, and the vascular lag maps will provide a useful contrast between the tumor and the healthy tissue. So the methods is an ongoing acquisition of 21 participants so far with different glioma characteristics. And the experiment paradigm is a breath hold test with 8 trials, in which it was a total of 20 seconds of apnea, exhale in before and exhale after the apnea. The data acquisition was 4 multi-echo sequence. We got an anatomical images of T1 and T2 flare and the expired CO2 signal. And for the data analysis, we used the data analysis explained in the Moya studies, where we pre-process the CO2 data with PTET and take the PET CO2 HRF signal. And also we did a multi-echo ICA decomposition using Sedana, which is an open source for multi-echo analysis. And I did a model classification using a visualization tool that is Blue. And there we use a FISU CVR, which is also an open source that we'll have a demo tomorrow explaining a little bit more, where we implement the lag GLM explain to obtain the CVR maps and the vascular lag maps. So for doing this, to explain our results, we have 4 cases, so you can see what we had. In this case, it's a glioblastoma grade 4 patient who did in the left side, we could see the task and the CO2 signal and the protocol that he had to follow. As we could see, he did a very good task. And we could see that exploration before the apnea and the exploration after the apnea was made. And in the right side, we could see the CVR maps, the vascular delay maps, and the T2 flare. And the CVR maps, you could see a small decrease if we compare it with the T2 flare. Let me see if you can, here, in this blue, showing that there is a decrease in the CVR. And in the vascular delay maps, we could see a much more information, which help us conclude that this is an informative lag and give us more information additionally to the CVR maps. In the case study 2 is a astrocytoma grade 3, which also did a good performance, as we could see in the left. And in the CVR maps, we had a decrease in the CVR areas of the tumor. And also in the vascular lag, we have more information of their lags or delays. So in this case, we classified it also as an informative lag map and a decreased CVR. In this case, we have an oligodendroglioma grade 2, like a low-grade glioma. Also did a good performance, and we could see that there's a decrease in the CVR. However, in the vascular delay map, we don't have very much information. We just can see a small increase of the delay in a small area. But we classified this one as a little informative lag. And for last, we have this case study where we also have a same class of tumor, oligodendroglioma, grade 3. We did a medium performance. We classified this as we could see that in the left side, in red, we have 2 missing breath holds that this patient didn't perform. And also we have an increase of the CO2 in the middle of the apnea, which indicates that expire before time. Seeing the maps, we see a decrease in the CVR map if we compare the left hemisphere with the right hemisphere. However, in the vascular delay maps, we didn't have any information. Also, in the CVR maps, we cannot see that the limiter of the tumor. We just can see that there's a decrease. So in this case, we got our 22 patients, we classified them. In 6 of them, we just had to discard, that are the red ones, because of a bad performance of task. In the yellow, we have the medium task performance patient that I explained before. In blue, we could see the patient who did a good performance, but had a good CVR map, but a little informative. They say some information, but not exactly what is happening from their tumor. And in green, we have a good task performance and also an informative lab. And here we can conclude that yes, the multi-echo fMRI is a feasible and a multi-echo fMRI breath hold task is a feasible and reliable task that can perform and give us CVR mappings in patients with glioma. Also, that we could have a reduced CVR maps value in glioma tissue if we compare it with healthy tissue, providing, yes, abnormal vascular behavior in the affected tissue. The vascular delay maps provides valuable insights and adding complementary information of the CVR maps. And it's important to take into account the potential differential diagnosis among patients, as each patient can present a unique result in terms of CVR and vascular lab. Thank you.